Hello, my name is Vasilisa. Welcome to my channel about brain research. I'm pretty sure that if you, at least once in your life, try to find any information about the brain, you found something about neurons, neuronal networks, and all other derivatives about neurons. But surprisingly, only the half of the brain consists of neurons. And all recent discoveries show that other cells play a critical role in everything that concerns brain, cognition, pathologies of the central nervous system, and etc. So today I'm very happy to introduce you Dr. Paolo De Rico, who is a specialist in microglia. And today we will talk with him about roles of microglia. So thank you for watching. Don't forget to subscribe and leave your like. So I have a question. So this is quite a typical question. I studied your CV, so it seems that you had a very smooth pathway from biology to neuroscience, but I'm pretty sure that science in reality is not so smooth. So could you please tell me how you came to neuroscience and to the topic you are working on now?
So pretty simple. So after my university study, I studied biology, normal biology, and then when I had to choose where to do a master thesis after my degree, I found a lab in Italy. So I'm from Italy, Milano, where they were studying so something more not 100% neuroscience, but sort of neuromuscular disease. So I was in between muscles and neuroscience. You know that the brain control every function, not only intellectual function, but also our movement, our muscles. And in particular, I was studying in a region of the central nervous system that is called spinal cord. And this is how I started. So I found this lab where I did my master thesis. Then I decided to continue doing a PhD, and my PhD program was a neuroscience program. So I continue studying this particular disease, neuromuscular disease, was a genetic disease. And then when I finished my PhD, since I already had a good background in anyway neuroscience field, I didn't want to change completely topic, so I decided to find another lab where I could continue the neuroscience. And then I moved to Germany, where I'm actually working also. And I found this lab where they are more focused on the brain. And this is how then I continue my study in neuroscience. And then when I moved here in Germany, I started to study Alzheimer disease. And then was something new also for me. So this interaction between immunology and neuroscience. And that's the reason, because then I started also to study microglial cells, or anyway, this immunity system of the brain. This is in brief my scientific pathway.
If you look at the banner of my channel, you will see the cells, and that are the cells from my master dissertation, and they are microglial cells. So, thank you, Paola. Could you please tell like for broad audience, what Alzheimer disease is and how it is related to the immune cells like microglia.
So I'm 100% sure that everyone at least once heard about Alzheimer disease. This is a really famous, unfortunately, really famous disease. This is a really is probably the adult neurodegenerative disease, not the most common neurodegenerative disease is related to dementia. And so this is a is considered a neurodegenerative disease because at the end the brain functions uh, are affected. So and this is due to accumulation in the brain to pathological proteins, so that are produced or in the brain, and they start to accumulate in the brain. So um, what they do, these proteins, so these are normal proteins, or not proteins that are normally expressed in the brain that have some function that are not yet completely understood. So what the normal protein do, but for some reason, these proteins start to become a little bit crazy and to fold in a non-physiological way, and then they start to accumulate in the brain, to precipitate and accumulate in the brain. This is one of the aspects of Alzheimer's disease. And then there is another hallmark of Alzheimer's disease, that is accumulation of another protein that is important in this case for um, the skeleton of the neurons. So these are produced internally by the neurons, and these are called tau protein. Also, in this case, there is overexpression of these protein that start to behave in a different way, in a normal way, and accumulate in the neurons. And this is considered really toxic and induce at the end the death of the neuron. So what happens at the end is that you start, depending, since Alzheimer disease start in particular regions of the brain. So for example, the hippocampus, where the memory is stored, or in the cortex. So Alzheimer has a regional now progression. And this is the reason, because the first sign of Alzheimer's disease is the memory loss, because one of the first region that is implicated in Alzheimer and is affected is this hippocampus, where the memory or that is important for memorization. And progressively, all the other regions are affected, and then of course when also the region of the brain that are important for living, breathing, eating, or I don't know, cognitive functions are also gone. There is nothing that we can do. But these are only the first hallmarks. But then there is a kind of feedback, positive, that implicate also other the involvement of other cells and other pathways or other mechanisms. And for example, when these kind of pathological proteins start to invade the brain, accumulate in the brain, this is recognized of course by our immune system of something wrong. So the immune system react in order to do something to help to remove these proteins that are not healthy. But then what happens is that they are not able to do, and this, for this reason, is enter in the game the microglia, because they start to remove what should be removed, but they are not able, and so they are chronically active, and this activation induces a sort of inflammation that at the end is also dangerous for the brain. So this is in brief what is happening during Alzheimer disease. Yeah.
Well, what do you think actually is the main target of the Alzheimer disease, or the main source of the Alzheimer disease, neuron or or probably some other cells like microglia that cannot cope with the accumulation of proteins in time.
So I can tell you when when scientists started to understand that microglia is really importantly involved in Alzheimer's disease, because, for example, we observe that there are specific genes that are expressed only by microglia that are mutated or alterated in Alzheimer's disease patients. So we observe that when there is specific mutation that affect microglia, the probability, so the risk factor to develop Alzheimer are increased. So when the scientists realize that there is a high correlation between microglia, also now the pharmaceutical companies start to become more focused on microglia. So in terms of drug developments or therapies, so people and scientists are trying to different approaches. So there are companies, for example, that are more focused on the neurons. There are companies that are focusing on producing drugs, for example, that can stop the production of these pathogenic proteins. Okay, blocking, for example, enzymes that can processing the production of these proteins, and many other people who are more focused on microglia. So this is our different approach. What I personally think is that we should have a more holistic way. So it's unfortunately, in particular in the last stage of the disease, is no more sufficient to target only one aspect, because there are really many other things involved. It's a little bit like a cancer. So of course there is a starting point, but then there are many other pathways involved, and so to try to target and cure only one aspect, it's not enough. Maybe at the beginning, yes. So this is the reason, because it's important to understand how the different stages evolve in the disease.
It was the same for the brain cancer. It was the same for the multiple sclerosis, for example, that now it's obviously that the neuron is not the starting point of the disease. And
Yes.
also all symptoms that patients established, they're actually marks of active state. So it means that the progression and the start of the disease happened much earlier. I found in the literature that estimated age of the start of Alzheimer's disease for men is about 35 years. So,
Yes. Yes.
and
That's
for women, it's later. And actually, do you have any explanation why we have this significant difference in dementia for men and for women?
Yes, but there is, so in terms of Alzheimer's disease, you are right. So it seems that the first sign of Alzheimer start earlier in men, but the percentage of females that develop Alzheimer is higher. And this seems to be related. We are not sure. We did also some study in my previous lab about this. Seems to be related to hormones. So we did a really nice study some years ago, where we wanted to see what are the progression of the pathology of, in particular, amyloid pathology in the brain in mice, comparing female mice who had multiple, I would say in English, pregnancy or litters. So they were multiple times. And we observed that more litters they had, so more times they were pregnant, and more aggressive was the disease. So it seems that, so I mean, with this, I don't want to discourage women to have children. Of course, it's something different between mice and women, but this is something that can be an explanation why females are more prone to develop Alzheimer's disease. Then males have other problematics in other dementias disease, but females, it seems to be some hormonal aspect also involved in the pathology.
I also found that in females it happens quite often that yes, women have dementia much later, for example, at the age of 60 or 70, and generally it happens after the start of menopause. But
Yes,
they have it, as you told, in more aggressive form.
yes, yes. And then we have to really to keep in mind that all the study that we do in mice, we are studying the genetic and the familiar form of Alzheimer's disease. That is the most rare, because normally, so 95% of the cases are, um, it's called late Alzheimer's disease. That is not a pure genetic disease. So it's induced by different factors, also lifestyle and so on. And only 5% of the cases have a pure genetic form. And these are the most aggressive. So the people who has particular genes that are mutated, for example, like ApoE, for example, or other genes.
These genes are involved in this amyloid beta production, right?
Uh, uh, so the ApoE is a protein that is important for the transport of lipids. There are different isoform of this protein. This is is a lipidic protein, so it can bind and transport lipids in the brain. This is important. I mean, and there is an isoform, a different form that increase the risk of develop Alzheimer. Also because there is a strong interaction and correlation between lipid production or sugar metabolism and Alzheimer's disease.
And
So
hormones,
this is
actually.
exactly, yes, yes. And that's, but I mean, and of course, all the study I was studying, all the study are mostly done in models that are genetically modified. So we push these mice or the animals to produce the pathology. That is not what is really is happening in 95% of the cases. But the point is that most of the animals are not developing spontaneously Alzheimer's disease. The mice don't develop, also because the point is that Alzheimer is a pure, so it's the higher risky factor is the age. So higher are the probability that the people get old and aged, and this is the case of today. Now we are extending the survival of people, but on the other hand, we are also extending the probability to increase age-related diseases like cancer, like dementia, Alzheimer. Uh, if you go behind of 100 years or even 50 years ago, 60 years ago, people died for other causes, okay, much younger, but there were not so many cases of Alzheimer's disease, for example, or cancer. Now there is
We,
this
we live longer, and now we start to have all these pathologies. Yes.
exactly, yeah.
So I would like to speak now about the implication of microglia in aging.
Hmm.
This is the, since this is the main risk factor for the majority of patients with dementia. But first of all, let's talk about origins of microglia and the life cycle of these cells. So these cells migrate to the brain
Mm.
during the early embryonic development, and
Yes.
when we don't have the totally formed the brain-blood barrier. This is the barrier that separates and defends our brain from pathogens. So generally it's formed between the astrocytes, this is other types of brain cells, and blood vessels, if we talk in brief. And tell me now, what happens with these cells that, for example, we are newborns,
Mhm.
uh, we have these microglial cells. Uh, what do they do?
Okay. So as you told, these microglia cells are not generated directly in the brain, so they don't have origins from this tissue that is called ectoderm, that is normally is the tissue that will form the central nervous system, but has the same origin of the blood cells, so they come from the periphery. And recently was demonstrated that these cells are formed in the yolk sac, in both in mice and also in human at the first embryonic stage. So they are formed in embryonic, and they have the same origin of the cells that will go and produce also the blood cells, right?
Mhm.
Um, so then at a certain point, these cells that are formed in the yolk sac migrate to the bloodstream and to the brain. Okay. There is also another source of these cells that is the liver. And so, of course, then we will not have any more the yolk sac, but only the liver. So this means that the cells that are then in the brain will start to maturate and differentiate in order to become the real adult microglia cells. And then there is a source of cells hematopoietic cells that are in the liver. And in case we need to replace the cells from the brain, these are also can generate new microglia cells that can migrate into the brain. But really briefly, so once in the first days after born, these cells are in the brain are completely different, so the morphology is completely different, but they are really, really important because, as you know, the brain of a newborn, but also children, is completely different by our brain or adult brain. And one of the main function of these microglial cells is what is called pruning. So what does it mean? So we born with much more structures of the neurons that are called synapses. So synapses are involved in the connection between neurons,
And
right? So
communicate.
and the
Yes.
communications, and this is extremely important because we learn and we memorize thanks to these synapses. So more synapses we have and more, but of course, so our brain work in a balance, in the right balance, in a so we are born with much more synapses, and microglia are important cells because they have another capacity. So they are also macrophages, so they can hit and eliminate structures that are not functional. So this means that uh, the neurons are generated, the synapses are generated in higher number, but then the synapses that are not functional, that are not important, they have to be in some way eliminated. And this elimination, this pruning, so microglia are like a freezer, no, like a tire cutter, so that they can regulate the number of these synapses. And then also these microglia are important because they are kind of let's say cleaner of the brain. So in the brain, there are cells that can die, for example, or cells that can be killed. And microglia are important because they clean and maintain clean our brain. So they remove all the dirty stuff that accumulate in the brain. This is the normal function of microglia.
Generally, people think that if they have more synapses or more neurons, it means that they will have better develop cognitive functions. But actually, biology shows the opposite picture. For example, in the case of some genetical diseases, when microglia do not perform this pruning or cutting all these structures and synapses that do not work during the first years of our life, it leads actually to cognitive decline in children. And what is
Exactly.
interesting for me, for example, how do microglia identify these neurons that will not work in the future? Do you
Mhm.
know some explanation of this?
Yeah. So what we think is that the microglia can um, sensor, so they can feel if a neuron is active or not. So you know that the neurons can communicate through electrical impulse. Okay. So when there is an electrical impulse in the neurons, the synapses that are the connection between two neurons release molecules, what is called neurotransmitters. And it seems that microglia can feel now if synapses is particularly active. So if the synapses release neurotransmitters, and in case the synapse is more silent, for example, microglia can recognize this and say, okay, this synapse is not working really well. This means that we have to eliminate or phagocyte or hit this synapses. This is the way how microglia, there are, of course, the mechanism are much more complex, but so the cells in general communicate each other, not only microglia, but also neurons and other cells communicate each other through sort of receptors that they express on their cells. And what is interesting that if you see, for example, an image or a time lapse or a movie of microglial cells, these are cells that have different branches or arms, and they continue moving. No, they are pretty static with their soma, but they continue extending and retracting their processes. And this during this time, they are continuously touching what is around, so neurons, for example, or
Mhm.
other cells. No, and with this extension and retracting, they can every time recognize what is happening around them, and this is so they are uh, yeah sensing the
Everything
environment.
permanent. Yes, in past people separated microglia, like talking about microglia in the resting state and the microglia in the active state. And nowadays we understand this that this is not a correct definition of microglia at all, because there is no rest for microglia at all. It
No.
they work permanently. And also these cells they modulate and start inflammation in the case of all pathologies that the nervous system can encounter. And what is interesting is that how do microglia, being like a best friend of the brain, how do they turn into a damage associated microglia? So this is the cells that are actually facilitate propagation of different toxins or toxic proteins like amyloid beta in Alzheimer disease. What did happen?
So the point is that so normally in normal condition you have a damage, for example, in the brain or in other region of your body. So normally you have this damage. This damage should be recognized by cells that are capable to solve the problem. So they come, they solve the problem. So in order to do this, there is a sort of inflammation. So the inflammation is the signal that is activated in order to tell, so look, there is something that is not happening well, so I need help. So the inflammation, so for example, if neurons or a region in the brain that is damaged, they release signals and they induce inflammation. So microglia, for example, comes and try to solve the problem, but in order to solve the problem, has to become a little bit stronger. So they became what we say, they became reactive. So they change completely their morphology, but they start also to produce other signals because if the damage is high and is serious, they need help of other cells. So they have to call help,
For
no,
other immune cells to
from
come
other
to
immune cells.
territory.
No, microglia itself is able to produce these inflammatory cytokines or signals in order to recruit other cells. And this is the normal situation. When then everything is solved, the situation go back to the normality. So that this is the important, and it's important also to don't take anti-inflammatory immediately when you have something, because the inflammation is something important. No, also the fever, for example, is a sort of inflammation. No, the problem is when microglia or these immune cells, because microglia are the first defense of the brain, the first line of defense of the brain, they are not able to remove the damage. In case of Alzheimer's disease, for example, now when there is a hyperproduction of these proteins that are pathological proteins, microglia try to do something, try to isolate these proteins that are not good, but in the meantime continue producing these inflammatory cytokines that at the end are dangerous. So if this inflammation start to become chronically,
Mhm.
this create the real problem. So that's the reason, because in normal situation, microglia are fundamental, are really important. But when something that cannot be solved is induced in the brain, in this case, or in other regions of also of the body, microglia is permanently active, and this condition is dangerous, the teleus, because then this continued production of pro-inflammation induce the death of neurons.
Thank you. But can astrocytes save the situation, for example?
At the meantime, also astrocyte became reactive. Yes, so all these cells that try to solve the problem, no, is this should be beneficial, no, but unfortunately, no, the most of the cases
At least
it's
at
not.
this stage, when we study the dementia and specifically Alzheimer disease, it seems that already both types of cells are impaired and do not perform well, right?
Yes, yes, true. But this is the reason, because of, for example, as you said, we start develop Alzheimer, for example, decades before, no?
Yes.
So I hope not, but could also have in my brain maybe some amyloid plaques. I don't know. I hope not, but can be. I mean, can be the case. And the point is that when you start having the first problem, and you go to the doctor, most of the time it's too late, because already the neurons are gone. And this is the reason, because all the drugs that now seems to work are drugs that are working at the onset of the disease, so at the beginning, the
Mhm.
first stages. But the point is that most of the people, they don't go to of course, to ask
You
for
don't know
specific
how to find these first stages.
exactly. So I would never go. So now, or when I have 30, go to the doctor in order to test if I have something wrong. No, right? So that's the reason, because it's also important in parallel to study also markers, so easy markers that can predict if you can develop Alzheimer, in order to start taking drugs, for example, or because at the moment, unfortunately, there is nothing that we can do. So the research is going ahead. So there are also different companies that are developing drugs that are working, but not in the late stages of this. Not when you have already the typical signs of the disease.
I also have a question about aging. So I wanted to say that nowadays it is known that microglia drives the aging of the brain with inflammation. And also women have the stronger inflammation in the brain they can live without dementia even, but this is just a fact, as I understood, right? That in the female brain, we have much more reactive microglia than in the brain of man. Is it true or it's a myth?
Active? What do you mean active? That they
Reactive. I mean this.
are reactive, or
Yeah, exactly.
this? To be honest, I'm not completely sure.
Mm-hmm.
Also because I mean it's really difficult to study microglia in human patients. So um, there are differences for sure. So now there are techniques that can compare in detail what are genetic differences or expression of different genes and compare male versus female. A lot is done on mice or in animal models. We don't know so much about human. There are differences, of course. There are genes that are differentially expressed by, but to say they are more active or not, it would be difficult. So it seems, so there is a study that is really interesting, is that they, but it's performed in mice, not in human. So they say that um, the microglia of males can be younger longer. So in females, we can discriminate three main stages of the disease that are clearly divided, can be clearly divided. In men, not. So it seems that the not men, males microglia stay younger longer, but then when it reach a certain point, they decline much worse than so the microglia of females seems to aged in a more would say in a more smoothie way.
Gradual way.
Exactly, yeah.
And by aging of microglia, do you mean that these cells become more reactive and more aggressive to environment? I mean,
They became more yes, they became more reactive, but they also lose some of the ability that normally they have. So one of the ability, for example, is the ability to internalize or phagocytose proteins or debris or the thing that can be this function to clean the brain.
Okay, so they still produce some active radicals, for example, and different molecules that damage surrounding cells or
Yes.
recruit immune cells into the brain, but they cannot clean these damaged regions. Very interesting. Thank you very much. But now let's talk about positive aspects of your about all these Alzheimer's studies. So I know that you are working on the electrical stimulations, and you study how these stimulations affect microglia and the brain of aging mice, I think, right?
Yeah. Yes.
Could you please tell about your study?
So what we are doing or trying to understand are the role of magnetic stimulation. So what is the magnetic stimulation? The magnetic stimulation is a non-invasive technique. So there is no pain, absolutely anything. So just apply close to the head of the patient a magnet in order to induce electric field in the brain. So the magnetic field can induce electric field in the brain, and it seems, in particular pathologies, but particularly in schizophrenia or depression, that this stimulation of the brain has a beneficial effect. Okay, it's not really clear what is happening, so that's the reason, because we are trying to study the effect. What we observe, for example, in terms of neurons, is that depending on which protocol of stimulation you use, you can have a potentiation of the synapses. So
Mhm.
the synapses can work better, or the opposite effect, because you have to consider, for example, that in Alzheimer disease, you have, so the neurons are more uh, excited, so they work too much.
Mhm.
Okay, it's like that's the reason, because, for example, Alzheimer patients, they develop easily epileptic seizure, no, because the neurons are too much active.
So
Okay,
they lack these inhibitory signals because
exactly,
in brain we have the balance of
exactly, yeah,
excitatory and inhibitory signals.
exactly. So the extremes are always not good. So you always saw that the brain, but not only the brain, in general, our body work to maintain no, a sort of equilibrium, sort of homeostasis, no. um, and this is what happening also in in the brain. So we observe that this magnetic stimulation can have a beneficial effect, or anyway some effect to
Mhm.
the synapses. What I'm trying now to understand, since there is a strong communication and involvement of microglia and neurons, I'm trying to understand whether also this magnetic stimulation can in some way influence the activity or other aspects of microglia. Um, yeah, so I'm trying to. I say I'm trying because it's really difficult, and the problem is that we don't know absolutely anything about this, and uh, we can really consider multiple aspects of microglia. No, we just in this time that we are talking, we talk about inflammation, we talk about phagocytic capacity, immune system. So you can really understand how many things microglia can do, and yeah, so to understand which direction take is not so easy.
You're working not in the mainstream. Generally, people, if they apply magnetic stimulation or transcranial stimulation, they actually stimulate neurons. So they
Mhm.
apply some energy to enhance or to decrease electrical activity of neurons. And
Yes.
now, you, as a person who is working not in the paradigm that the neuron is the center of the brain, you use the same method, but you are looking at microglia. So it is not typical, and bravo.
Yeah, I mean, it's also high risky, because so we don't really know what can happen. I cannot exclude that the alteration in neurons can also indirectly induce something to microglia. This I at the moment we don't know.
So good luck with your research.
Thanks.
Also, since you're working not in the mainstream, did you meet some rigid professors that didn't take seriously probably this separation, um, the separation of microglia, for example, on the different stages, or people that just do not believe that microglia are very important in Alzheimer disease, and that these cells uh, can be the pivotal cells in this
Mm.
disease.
So this was probably true in the past.
Mhm.
So consider that people started to be really interested in microglia field uh, since 20 years, more or less, 15, 20 years. Before, microglia were not really considered. Uh, but now, no, the interest is increasing a lot, and there are more and more studies describing the role of microglia, because microglia is involved in all the neurodegenerative diseases. So microglia.
Exactly.
So this is something that, of course, as I told, focusing only on microglia, believing that only targeting microglia can change the situation, I'm not really so positive and confident in this. Can also be, I don't know, but for sure is one of the main players in different diseases, not only neurodegenerative diseases, so where you have more physical aspect, but also in cognitive disease or in, I don't know, other problematic like autism or depression or schizophrenia,
Or
um,
multiple sclerosis
multiple sclerosis. Yes, yes.
or
But
brain cancer. Yes.
but a common aspect is in all the neurodegenerative diseases we see reactive microglia.
Do you think that we have the progress in treating neurodegenerative diseases?
We have a lot of improvement. So in particular, we know a lot now, really a
Mhm.
lot of things. So with this new technique where we can really, at a single cell level, we can see what is happening at transcriptional, no, to see how much the genes are expressed. We really can know many things. We are far away to find a cure, I have to be honest, but something is moving. There are more and more drugs that are, so they are promising drugs, but they are mostly working at the beginning of the neurodegenerative disease. And then of course, so all the diseases are really different, and it's not possible to design something that can work for all the neurodegenerative diseases. So every disease should be, and also for each in the same disease, for example, Alzheimer, we can have many different aspects. Because, for example, another bad thing of Alzheimer is that different diseases can coexist in the brain. And this is really problematic because then you don't know how you can treat these. But of course,
Especially if we're talking about age-related diseases, exactly.
yes, of course. Yeah.
Thank you very much, Paolo. It was a pleasure for me to discuss these very tricky topics. And the final point, could you please find 3 words that help you to do your research, that led you to science, what actually drives you ahead and inspires? 3 words.
It was bad. Curiosity is the first, of course. Then open mind is something really important. And then be patient. So I think this is the most important quality, because so of course the people think that now science and to do research is nice, and yes, of course it's nice, but it's really can be really frustrating. So the results can be obtained most of the time also by, you have to be lucky to say, but you have to be really patient, because maybe you don't obtain anything or you don't see anything for 1 year, and then you do the right experiment and that will change everything. So these, I think, are the 3 main, at least the most important thing for me.
Thank you very much, Paolo. So keep calm and take care about your microglia.
Of course, this is really important. Yeah.
So thank you for watching this episode. If you liked my video, so leave your like, subscribe. If you have some questions, leave a comment. See you.